Hello everyone, and welcome to Pearson Bio Academy. I am Fazna Imran.
And this is the place where you can top up your biology knowledge with our past paper explanation through very clear and short video series. And currently we'll be focusing on Pearson Edexcel International Advanced Level, paper reference WBI 1101, Unit 1, which covers molecules, diet, transport, and health. This is part 2 of my carbohydrates video series. If you haven't watched part 1 yet, I highly recommend that you do so first, as it includes a brief course introduction. In this video, we'll quickly revisit the introduction to ensure we are all on the same page. As you can see on the screen, there are 6 key topics in Unit 1. If you need a detailed overview, you can find it in part 1 of this series. I will include the link in the description box below. As you may know, I have created further categorization under the chemistry of life. In this section, we will explore questions related to carbohydrates, the chemistry of water, proteins, lipids, and enzymes. And right now, our focus is on carbohydrates. We have already completed part 1 of the carbohydrate series, which covered disaccharides, and in part 2 we will tackle questions from 2020 to 2024, specifically focusing on polysaccharides. Let's start by examining how polysaccharides are formed by joining multiple monosaccharides. As we learned in the first video, the formation of a glycosidic bond occurs between two monosaccharides. This process is important in creating a large polysaccharide as well, which also contain glycosidic bonds. So there are two main types of glycosidic bonds, 1,4-glycosidic bonds and 1,6-glycosidic bonds. First, we'll focus on the 1,4-glycosidic bonds. In this process, a water molecule is removed, allowing two monosaccharides to join together via 1,4-glycosidic bond. As you can see, this mechanism continues with each additional bond formed. Every time a monosaccharide is added, a 1,4-glycosidic bond is established, leading to the growth of the polysaccharide chain. And this is the fundamental way that polysaccharides are synthesized from multiple monosaccharides. During the formation of a 1,4-glycosidic bond, you will notice that the polysaccharide chain is linear and straightforward. In contrast, when we refer to 1,6-alpha-glycosidic bond, we are talking about branching within the polysaccharide structure. This bond occurs between the first carbon of one glucose molecule and the sixth carbon of another. And this branching is important because it adds complexity and variation to the polysaccharide structure. The ability to form branches is essential for the functionality of many polysaccharides, as it influences their properties and how they interact in biological system. Understanding these types of glycosidic bonds is key to grasp how polysaccharides are structured and how they function. And the first question is from October 2020 paper. So here the statement reads, glucomannan is a dietary supplement claimed to aid weight loss. So this glucomannan is a new term to you, but the question, glucomannan is a branched polysaccharide similar in structure to amylopectin. So we will later discuss about amylopectin. However, they are asking about branches. So which glycosidic bonds are responsible for the branching in glucomannan? You know it's 1,6 only. In the next question, you will find a table where you need to mark crosses in each box comparing monosaccharides and polysaccharides. The first statement have the general formula CnH2nOn, and you know this formula only applies to monosaccharides because when forming a polysaccharide, a water molecule is removed each time, and this formula therefore does not apply to polysaccharides, so this is only matching for monosaccharides. The next statement have glycosidic bonds. You know glycosidic bonds are formed during the bonding of monosaccharides to create polysaccharides. Therefore, this characteristic is unique to polysaccharides. And the third one have little effect on water potential. So this statement addresses how each type of carbohydrates interacts with water. Monosaccharides dissolve completely in water, therefore they significantly affecting water potential. In contrast, the dissolution of polysaccharide in water is limited, therefore resulting in only a minor impact on water potential. Therefore, this is matched with polysaccharides only. And on the polysaccharides, you have glycogen and starch. First, we will start by examining glycogen. This is the structure of glycogen. You can see the both 1,4-glycosidic bonds and 1,6-glycosidic bonds are there. So therefore, they are branching structures. First, let's look some general facts about glycogen. So they are made up of alpha glucose, and they are storage molecule in human and fungi, and they have both 1,4-1,6 glycosidic bonds. And the first question from January 2021 paper, humans store energy as glycogen, and you have to find out the correct statement about the formation of glycogen. And you know they are made up of alpha glucose molecule, and you know they are joined together by a condensation reaction. So during the condensation reaction, one H2O molecule will be removed. Therefore, the correct answer is A. Alpha glucose molecules join together by a condensation reaction. The second part of that question, name the bond that joins two glucose molecules together. It's glycosidic bonds. The next question is from the same paper, and it asks you to explain how the structure of glycogen relates to its role as an energy storage molecule. And this question you need to justify how the specific structure of glycogen helps it perform its function efficiently. First, we will look some key points. You know, glycogen is made up of many glucose units, and glucose are the fuel for ATP production. So since it consists of many glucose units, it has a high energy content. And next, glycogen is insoluble in water, which means it does not have an osmotic effect. So this is advantageous because storing glycogen in liver cells and muscle cells prevents any osmotic imbalance within those cells. And another important feature of glycogen is its branched structure. This branching allows for quick energy release when energy is needed. The cell can break off a branch and give glucose rapidly. And finally, despite being a large molecule, glycogen is very compact. So this compactness means it has a high energy density, allowing a significant amount of energy to be stored in a relatively small space. So in summary, glycogen is polysaccharide composed of many glucose units, and therefore its high energy content. They are large and they are insoluble structure and branched form for quick energy release and compact nature all contribute to its effectiveness as an energy storage molecule. So you can give your answer like this. Glycogen is a polysaccharide composed of many glucose units, therefore it has high energy content. Its large and insoluble structure make it unable to dissolve in water, thereby preventing any osmotic effects within cells. The branched structure of glycogen allows for rapid breakdown and quick energy release when needed. Additionally, its compact form provides a high energy density, allowing cells to store a large energy in a relatively small spaces. The next question is from the January 2021 paper. It involves a small calculation and they may present you with a new concept. However, don't worry, just read the question carefully. This question states that von Gieck disease is one type of glycogen storage disease, abbreviated as GST. And between 1 in 20,000 and 1 in 25,000 babies are born with GST. About 25% of patients with GST are thought to have von Gieck disease. And in one country, 3.8 million babies were born in 1 year, and we have to estimate the number of babies born each year with von Gieck disease in this country. First, we need to estimate the number of babies with GST, as this is the range provided. The upper limit of the range is 1 in 20,000. So to find this, we divide 3.8 million by 20,000 and it's 190. Next, we find the lower limit of the range, which is 1 in 25,000. We calculate this as follows and it's 152. So thus the range for the number of babies with GSD is 150 to 190. We can estimate the number of babies with von Gieck disease from this range. Since 25% of GST babies are thought to develop von Gieck disease, we can take an easy number for calculation. Let's use 160. You can pick any number in between 152 to 190. To find the number of babies born with von Gieck disease, just you have to multiply these 25% with the number of babies with GST, and using 160 as our estimate, we have the number of 40. So if 3.8 million babies were born, we estimate that about 40 babies may develop von Gieck disease. And according to the marking scheme, the examiners will accept any value in between 38 and 50. So you can choose any number within that range. Additionally, the examiners have been instructed to accept non-whole numbers for this question. So don't worry about needing to provide a whole number. The next question is from the June 2021 paper and it's a straightforward question. As usual, they have provided a little bit additional information before the question. A person with diabetes has a blood glucose level that can be too high. When the blood glucose level of a person without diabetes becomes too high, the liver stores glucose as a polysaccharide with the help of insulin hormone. And the question is, which polysaccharides does the liver store? The answer is very simple, it's glycogen. The next question is from the 2022 October paper. Again, they provide some information first. The statement reads, insulin lowers the levels of glucose in the blood. This glucose is taken up by the liver and stored as glycogen. The question asks you to compare and contrast the structure of glucose with the structure of glycogen. So in this case, we are comparing a monosaccharide with a polysaccharide. First, let's recall the structures of glucose and glycogen. So you can see both share some similarities and differences. As you can see, both glucose and glycogen contains the same elements, carbon, hydrogen, and oxygen only. And also they both contain alpha glucose. However, you can see glycogen is made up of many alpha glucose units. And if you look up the differences, the structure of glycogen is very large, consisting of many glucose molecules, while glucose is a single molecule. And also, glycogen contains both 1,4 and 1,6 glycosidic bonds, while glucose has no glycosidic bonds as it is a monosaccharide. So you can summarize your answer like this. And please make sure if they ask you to write, compare and contrast to give your answer in a paragraph form, not in a bullet forms. So here the answer. Both glucose and glycogen contains the elements carbon, hydrogen and oxygen only. They are made up of alpha glucose, but glucose exists as a single alpha unit serving as a monosaccharide. Glycogen is a complex polysaccharide composed of multiple alpha glucose units linked together. Glycogen also has both 1,4 and 1,6 glycosidic bonds, whereas a single glucose molecule does not have these bonds. So always you have to write your answer in your own words, making a paragraph. Now let's take a look at starch molecules. They are composed of alpha glucose unit and they serve as energy storage in plants and consist of both straight chain amylose and branch chain amylopectin. And here is a diagram from your textbook showing amylose and amylopectin. You can see the differences between them. In amylose, there are no 1,6 glycosidic bonds, so there are no branches and it tends to form a spiral shape. On the other hand, amylopectin has both 1,4 and 1,6 glycosidic bonds which allow it to have branches. The first question related to polysaccharide molecules comes from the January 2022 paper, specifically from the first question. In this question, you are provided with a photograph showing a saffron crocus plant. The question states, this plant grows from a bulb. The bulb contains starch as an energy storage molecule for the crocus. In plants, different organs can store energy, and in this case, it's the bulb. The question asks, how many of the following statements are correct for starch in living cells? So let's go through the statements one by one. The first statement is that starch consists of a mixture of two types of polysaccharides. This is straightforward and correct because starch is made up of amylose and amylopectin. The second statement claims that starch contains only 1,6 bonds, which is incorrect. Starch consists of both amylose and amylopectin, and amylose has 1,4 glycosidic bonds, while amylopectin contains both 1,4 and 1,6 glycosidic bond. Therefore, starting it has only 1,6 bond is incorrect. The third statement reads, starch is insoluble in water, which is correct. Starch is less soluble and generally considered insoluble in water. Therefore, the third statement is correct. So, two statements are correct here and it's answer C. The next question is from the October 2023 paper, a very recent past paper question. It provides some information about starch, stating that starch from plants is used in the production of food, paper, and building materials, and the structural and chemical properties of starch depend on its amylose and amylopectin content. You are given a table to complete focusing on the types of bonds. So first we will look about the glycosidic bonds. And before we proceed, let's revisit the diagram. So as you can see, both amylose and amylopectin have glycosidic bonds between their glucose units. So the correct answer here is that both amylose and amylopectin have these bonds. The next statement is 1,6-alpha bonds. So recently papers have stated specifying these as 1,6-alpha bonds because they form with alpha glucose, and your textbook mentioned them as 1,6-glycosidic bonds. So, but in the exam, they have been modified to 1,6-alpha bonds to emphasize the involvement of alpha glucose. So, moving forward, when referring to 1,6 glycosidic bonds, you can specifically call them 1,6-alpha bonds. And these bonds are found only in amylopectin, so the correct answer is amylopectin only. And finally, we have hydrogen bonds. So these occur when there are hydroxyl group leading to slight positive and negative charges, which allows the formation of hydrogen bonds. So both amylose and amylopectin contain hydroxyl groups, so hydrogen bonds can form between the molecules. In this picture, you can see the formation of hydrogen bond. So therefore, the correct answer is the both amylose and amylopectin have hydrogen bonds. This is a schematic diagram from your textbook showing amylose, amylopectin, and glycogen. If you can keep this picture in your mind, it will help you easily recall their structures. As you can see, amylose is a straight chain molecule. They contain only 1,4-glycosidic bonds. And amylopectin, on the other hand, has branches, so which means it contains both 1,4-glycosidic bond and 1,6-alpha bond. And similarly, glycogen also has a branch structure with straight chains, and also they contain 1,4 and 1,6-alpha bonds. The next question is from the same section, October 2023 paper, and this appears to be very important question related to starch molecule. In this question, you are provided with a graph illustrating how when different sources of starch are heated in water, amylose moves from the starch source into the water. So the graph shows the percentage of amylose lost from 4 starch sources at 3 different temperatures. By analyzing the graph, you need to answer the questions below. First, let's examine the graph. On the x-axis, you have the source of starch, and on the y-axis, you have the percentage of amylose lost from each source. Looking at the data, you can observe that as the temperature increases from 60 to 70 and then to 80, the amount of amylose lost also increases across all 4 starch sources, for potato, and for cassava, and for sweet potato and maize. And the question asks for conclusions regarding the effects of temperature and the source of starch on the loss of amylose. So if we analyze the data, we see that the loss of amylose increases with higher temperatures for all sources, And additionally, you can say the highest loss of amylose is consistently seen in cassava at all temperatures. So the question is, describe two conclusions that can be made about the effects of temperature and source of starch on the loss of amylose. So under effect of temperature, you can write like this. Along with temperature increasement, the loss of amylose increase in all four different sources. And if you talk about effect of source of starch, you can see the most amylose lost is from cassava at all three temperatures. We'll be moving to the C part. So here they have provided you a small information. The loss of amylose and amylopectin during the cooking of rice affects the hardness and stickiness of the cooked rice. The effect of temperature and the loss of amylose and amylopectin was investigated. So here we are going to investigate how the temperature going to impact the loss of both amylose and amylopectin from the rice. The diagram shows part of the method used. First, let's take a look on the steps. The first step, they have mixed the rice with water and soaked for 30 minutes. Then they have divided the mixture into 9 equal samples, and in the third step, they have heated the different mixture at a different temperature, and finally when the liquid reached the desired temperature, they have separated the liquid. And finally, the liquid collected at each temperature was analyzed for amylose and amylopectin. Now let's provide a schematic view for this practical. We have rice and we'll begin by adding water to it. The rice will soak for the first 30 minutes. After soaking, we will divide the rice into 9 equal parts using test tubes. It's always better to label them for clarity. Now let's take a look at each test tube. This is how they will appear after we have prepared them. The rice contains both amylose and amylopectin. After heating each test tube, some amylose and amylopectin will move into the liquid, and we will analyze this liquid part. By measuring the percentage of amylose and amylopectin present in the liquid, we can deduce the amount that has been lost from the rice. In this investigation, we have a graph showing the results with temperature on the x-axis and the concentration of amylose and amylopectin in the liquid on the y-axis. So before we analyze the graph, it's helpful to consider the underlying question, as this will guide our analysis and make it easier. The question asks us to compare and contrast the effects of temperature on the loss of amylose and amylopectin from rice. Now that we understand the goal is comparing the loss of amylose and amylopectin, so we can proceed to analyze the graph directly. So this approach allows us to use our time more effectively and gain a clearer understanding of the data. When we closely examine the graph, we can see that the loss of both amylose and amylopectin increase with temperature, but it's only up to 85 degrees Celsius. And beyond this point, both compounds begin to decrease. And also you can see the highest loss of both amylose and amylopectin occurs at 85 degrees Celsius. And also at approximately like a 47 degrees Celsius and around 64 degrees Celsius, the loss of both amylose and amylopectin is equal. However, when we look at the overall trend, you can see that amylopectin losses more than the amylose above 64 degrees Celsius. And also you can note this one also. The amylopectin starts to be lost at very lower temperature, and while amylose begins to lose there at around 40 degrees Celsius. So you can write the answers like this. The loss of both amylose and amylopectin from rice is increased with temperature up to 85 degrees Celsius, after which both decrease. And the highest loss of both amylose and amylopectin occurs at 85 degrees Celsius. Notably, loss of both from the rice is same at 47 and 64 degrees Celsius. Overall, more amylopectin is lost than amylose above 64 degrees Celsius, and further, amylopectin starts to be lost at a lower temperature, while amylose begins to lost at 40 degrees Celsius. Here I have given you a complete answer, but you will score if you write 3 points, at least 3 points, which is matching with the scheme. In the next part of the investigation, a replacement liquid was added to complete the cooking of the rice. As you know, heated during cooking causes amylose and amylopectin to leach into the cooking liquid, and in this investigation the researchers changed the cooking liquid and investigated the hardness and stickiness of the cooked rice, and the results are shown in the table provided. And when you are comparing the hardness and stickiness of cooked rice, we need to determine which component contributes to each property. So looking at the hardness of cooked rice, we see that the highest value is 27.65, which correspond to the water containing both amylose and amylopectin. And additionally, the liquid containing only amylose has a similar value, suggesting that both the liquid containing amylose and the one containing both components contribute to the hardness of cooked rice. And for the stickiness of cooked rice, we can identify 2 key points. The water containing amylopectin and the water containing both components contribute to the stickiness of the rice. So to summarize, the hardness of the cooked rice is determined by the presence of amylose, while the stickiness is influenced by the amylopectin. Now let's address the question. In this question, we ask to comment on the effect of the replacement liquids on the hardness and stickiness of cooked rice using the information provided in the table. So let's begin by summarizing our key findings. Overall, the data suggests that the rice cooked with water containing both amylose and amylopectin has both higher hardness and stickiness. And the water containing amylose contributes to the hardness of the cooked rice, while amylopectin contributes to the stickiness. And now let's discuss an important aspect often overlooked: the need for standard deviation and error bars. Standard deviation provides information about how much the measurements vary from the average value. If the standard deviation is very high, it means there is a lot of variation and the results may not be consistent across multiple trials. For example, just imagine you are doing 2 experiments to measure the hardness of cooked rice, and one shows hardness value of 27.65, with a standard deviation of plus or minus 5. And while another shows 27.65 the same with a SD of plus or minus zero point 0.5. So even though the average is the same, the first experiment's wide variation indicates inconsistency, while the second is more precise and reliable. And without knowing this variability, we can't confidently say that the observed trend will always be true under similar conditions. And when drawing conclusions from the data, we need to ensure they are dependable. So without standard deviations and error bars, we can't determine whether the observed differences are consistent or if they might fluctuate significantly. And this makes it hard to generalize the findings. So if you would like to see more videos on how to analyze graphs or dive deeper into the statistical aspects, especially those frequently appearing in your question paper, please let me know by leaving a comment below. The last part from this question suggests why rice should be cooked in a measured volume of water with a lid on until all the cooking liquid has been absorbed, and you have to use the information in the table to support your answer. So for this question, we need to consider two aspects using a measured volume of water and cooking with the lid on. So first we look why we have to use the measured volume of water. So this ensures that all the liquid is absorbed by the rice during cooking. If you add too much water, some may escape, causing more amylose and amylopectin to be lost. So by controlling the amount of water, you can maintain the right balance of these components. Since amylose contributes to hardness and amylopectin, they controlling the stickiness of the rice. Therefore, controlling the liquid helps achieve the desired texture of the cooked rice. And cooking with the lid on, this prevents water from evaporating, ensuring that the rice absorbs all the liquid. So this way, the rice cooks evenly and you can maintain the right balance of hardness and stickiness by keeping amylose and amylopectin in the cooking process. So finally, you can give your answer like this. Since it's a 2-mark question, this would be much enough. So cooking rice is a measured volume of water with lid on ensures that all the liquid is absorbed by the rice during the cooking process. Therefore, minimal loss of both amylose and amylopectin, resulting in a well-cooked rice that meets desired hardness and stickiness. I have highlighted the main points. You can understand it very well. The next question is from the January 2024 paper and it focuses on enzymes. The statement reads, amylase in saliva breaks down amylose into maltose. If you've studied the digestive system, you'll know that the chemical digestion of amylose starts in the mouth. The question asks, which row of the table describes the action of amylase? So we learned in earlier slides that enzymes help break down polysaccharides through a process called hydrolysis. And this was also explained in part 1 of the carbohydrate video. So during hydrolysis, water is used to split the polysaccharide. Specifically, amylose is broken down by breaking its glycosidic bonds. Amylose contains only 1,4-glycosidic bonds. And these are the bonds that amylase targets and breaks. So therefore, the correct answer is hydrolysis, and the type of bond broken down is the 1,4-glycosidic bond. So the final answer is C. The last question is from the 2024 May paper. So if you look at this question, it connects closely to the previous one from the January paper which we discussed earlier. Earlier, they introduced a short question, and this time they expanded on that concept, creating a larger main question. So this is a great example of why learning past paper is essential. Often a smaller part from one paper gets expanded into a more detailed question later on. The statement provided states starch is a polysaccharide composed of amylose and amylopectin. We know that. Digestion of starch begins in the mouth by an enzyme called amylase. And this amylase can break only 1,4-glycosidic bonds. And only disaccharides and oligosaccharides are produced during this reaction. You know, oligosaccharides mean the molecules that contain more than 3 monosaccharides but less than 10. The diagram in the question shows part of an amylopectin molecule. The colored circles indicate glucose units joined by 1,4 and 1,6 glycosidic bonds, while the white one or the non-colored one shows glucose linked only by 1,4 glycosidic bonds. And the question asks you to do 1 disaccharide and 2 different oligosaccharides that could be produced following the digestion of this part of amylopectin. So when we break down amylopectin, we must remember that we cannot separate 2 colored monosaccharides because they are linked by a 1,6 glycosidic bond. So amylase cannot break this type of bond, therefore any disaccharide produced will still have these 2 colored bonds connected together. So there is a possibilities of disaccharides like this one, the colored one, or any non-colored or colored joined together. When discussing the formation of oligosaccharides from amylopectin, it's important to remember that the same concept as with disaccharides. You cannot separate the colored ones because the glucose units connected by a 1,6 glycosidic bonds. And therefore, these 2 monosaccharides will go to the oligosaccharides together. So there are some possible ways. And you can draw one of them. So you have to make sure that these oligosaccharides contain more than 3 to lesser than 10. And the second thing is the 2 colored one should be go together. The next questions ask you to explain why this amylase is able to break only 1,4-glycosidic bond. Actually, this question is regarding the enzymes, but I have brought here. And we will be repeating this question again under enzyme series also. So this is due to the specificity of the amylase enzyme. You know, enzymes are specific to their substrates, so meaning that amylase can only bind to the 1,4-glycosidic bond because its active site is shaped to fit that specific bond only. Therefore, amylase can break down only a 1,4-glycosidic bond, not the 1,6 glycosidic bonds. So you can write the answer like this. Amylase enzyme is a specific and it can only bind with 1,4-glycosidic bond because only 1,4-glycosidic bonds can fit into the active site of this amylase enzyme. The next question states that the digestion of carbohydrates is completed in the small intestine, where glucose is then absorbed in the bloodstream. While starch digestion begins in the mouth, it doesn't fully break down carbohydrates into their monosaccharides. Instead, further digestion occurs in the small intestine. The question asks how disaccharides and oligosaccharides are broken down in the small intestine, and you can relate this to the previous question. We discussed how amylase can only break down 1,4-glycosidic bond, resulting in the formation of oligosaccharides and disaccharides. And in the small intestine, they might have some specific enzymes that can hydrolyze different types of glycosidic bonds, including 1,6 bonds present in oligosaccharides and in between disaccharides. So these enzymes are tailored to break down these specific bonds, allowing for the complete digestion of carbohydrates into monosaccharides for absorption into the bloodstream. So you can write your answer like this. Disaccharides and oligosaccharides are broken down in the small intestine by different enzymes which is specific for each type. These enzymes hydrolyze the different types of glycosidic bonds present in each sugar. That's all for today's video. I hope you found this explanation helpful. If you did, make sure to hit the subscribe button and turn on notifications and you don't miss any future videos. Don't forget to like, comment and share with your friends who might find this useful. Thanks for watching and see you in the next one. Bye bye.